Continuing with enzymes. Okay, so enzymes, there is a classic question that comes up all the time. In fact, it's more of a protein question. It's most commonly asked as an enzyme question. So I'm going to call it the protein shape question. Now, this could be enzymes, which it most commonly is, but it could also be antibodies. Remember, we've got EATS, E-A-T-S, enzymes, antibodies, transport, and structural proteins. They're the different types of proteins. So this is going to be now the key terms are going to go in red, tertiary structure of now what's the important bit within a protein or an enzyme? It's the active site, or it would be the variable region of an antibody, is complementary to the shape of the substrate and forms something that needs to be in all of your answers when you're talking about any of these things, an enzyme substrate complex. Quite commonly, this is written as an ES complex. This could also be an antibody-antigen complex, depending on the type of molecule. Now, this is for a functional, obviously this forms an enzyme substrate complex, so it's working, it is functional. What about you get a question, it's asking you why a protein is no longer functional? So non-functional protein. Again, this could easily be an antibody. Now, there's two reasons why this might be non-functional. The most common is going to be a mutation. Now, if it's a mutation, we're going to look at this in a lot more detail when we do the science behind it, but this changes the primary structure. So that's the sequence of amino acids. And remember, we can write primary as one like this. The primary structure of the protein, and if you change the primary structure, this changes the tertiary structure of the protein. And now, actually, the rest of this is going to be the same as our answer from over here. So, if the enzyme is denatured, remember that's the temperature causes the molecules to vibrate so much it changes the internal bonding. So this changes the ionic bonds, which changes the tertiary structure. And now both of these things, we've now changed the tertiary structure. So now we can continue for both of them the same. This changes the shape of the active site. Now we can see a pattern emerging. We've changed the shape of the active site. The substrate, excuse my handwriting, the substrate is not complementary anymore. And then finally, what do we need to say? We always need to mention the enzyme substrate complex. So in fact, I'm going to put it in capitals, no enzyme substrate complex. And that's why it's non-functional. So you need to think with these questions. First of all, is it functional or not functional? The answer has got very much the same structure. First of all, think, is the primary structure relevant? Has the sequence of amino acids changed, yes or no? If the sequence of amino acids has changed, then you need to mention the primary structure. That's generally a result of a mutation. The change in the structure of the primary structure is going to cause a change in the tertiary structure. So we can write tertiary like this. We can also write tertiary with 3'. And then we need to say, is the substrate complementary to the active site? So we always have to mention the active site, and we always need to say if it's complementary or not. And then we need to say, finally, does it or does it not form an enzyme substrate complex or an antigen-antibody complex or antibody-antigen complex? If you can answer the questions using that terminology in that structure, you will get full marks on all those enzyme questions.